Our understanding of how we can alter our own lifestyles to reduce the risk factors associated with cancer is much better understood. A healthy and balanced diet, an active lifestyle, and reduced intake of carcinogens such as cigarettes, drugs and alcohol all correlate to reduce cancer development and progression. However, we can't completely abolish our risk of developing cancer. Due to the genetic mechanisms that are random or inherited, we still don't fully understand this disease. By improving our understanding of these mechanisms on a molecular level, we can unlock the potential to produce efficacious and therapeutic alternatives for cancer patients worldwide. Today, I'll be discussing the long non-coding RNA H19 and how we can leverage its impact on cellular processes to prevent the development of cancer. Cancer can develop in all organs of the body. However, survival rates are particularly low in metastatic tumours. This is where a primary tumour comes from one organ and is delivered to another organ to set up a secondary tumour. Metastatic development requires a process called epithelial to mesenchymal transition, or EMT. What is the epithelial to mesenchymal transition, and why is it so important in cancer? This is a process by which epithelial cells lose their cell-cell adhesion and polarity and transform into cells of mesenchymal properties. The balance between these two cell states is critically important in cancer development. Here's a diagram to make this a bit more clear. Here we can see a clear contrast in the two cell types. On the left, epithelial cells, which are more rounded and clustered. Overexpression of these can result in proliferation and tumour development. Whilst on the right, we have the mesenchymal cell type, with elongated cell bodies. Overexpression promotes an invasiveness of cells. We can also see this in vitro. To find out a bit more, let's look closer. Wow! As you can see, we're in the bloodstream now. For tumour cells to form metastatic tumours, they have to transport into the bloodstream and to the distant site to form the tumour there. Cells from tumours travel through the basal membrane into blood capillaries or lymph vessels, travel to their distant site, and there they exit and go into the surrounding tissues. Here, they have to revert back to epithelial phenotypes to make sure that they can proliferate and grow as new tumours. Scientists call this intravasation and extravasation in cancer cells. Let's have a little look further inside. Looks like there are viruses about. Better keep moving. And this is where the magic happens. We're now in the nucleus of the cell. Behind me, we can see a large strand of DNA. This is what initiates the process of transcription and translation, which results in the formation of proteins. Transcription is the process whereby the alpha helix of the DNA unzips, forms two separate strands, and then complementary bases attach. These join together to form a new RNA strand. The next process is translation. Here, messenger RNA at ribosomes forms amino acids which turn into polypeptide chains, which in turn form proteins. These are essential in building our cells, tissues, and organs. Okay, I think it's time to get out of here. Whew. Thought I'd get some fresh air after that. So we've now seen how RNA can mediate the process of converting DNA all the way to protein via transcription and translation. However, a recent study called the GenCode study revealed that the human genome is far more complex than we once thought. This study revealed that whilst there are over 20,000 protein coding RNA sequences, there are even more RNA sequences that don't code for any protein at all. These can control the regulation of the coding RNA from RNA to protein. H19 is one of the key non-coding RNAs, and it can have a control on both EMT and eventually cancer. So, we now know that EMT is not only important in our normal development, but when it's misregulated, it can lead to cancer and tumour development. If we can increase our understanding of the molecular regulation behind this process, then we can generate effective treatments. There's now a wealth of evidence that H19 has a role to play in EMT. Let me show you a bit more. H19 is frequently overexpressed in cancer, targeting transcripts which regulate normal EMT to misregulate the process. Here we see one brain tumour study and one colorectal tumour study, showing an increase in H19 results in an increased tumour size. So there we have it. H19 is an essential part of cancer development.
Okay, but what else can be done to cure cancer?
So there are three main areas we need to understand more. One, we need to discover more long non-coding RNAs that are involved in cancer. 2, we need to characterise the full set of H19 and other molecule interactions between normal cell, tumour cell and metastatic tumour. And 3, we need to develop mechanisms to safely and specifically deliver these molecules into the body. Once we've done this, we can be hopeful of increasing survival rates of cancer worldwide.